Hello everyone, welcome to this CNH Cognitive Neuroscience Hub seminar. So before I start, I just want to acknowledge the traditional owners of the unceded land on which we have this seminar today and where we're working and learning, the Wurundjeri Wurrung and Wurrung people. And I want to acknowledge the fact that they've been taking care of the land and kind of transmitting culture over the years. Now, I'm very happy to introduce Dr. Frini Karayanidis for our seminar today. And she's a professor of psychology at the School of Psychological Sciences at the University of Newcastle and the director of the university's functional imaging lab there. Professor Karayanidis did her PhD at the University of New South Wales before completing postdocs at Macquarie University in Sydney and the Hospital Centrist in Montreal before taking a position at the University of Newcastle. Professor Karayanidis leads a multidisciplinary research program aimed at developing accessible composite vascular and neural brain biomarkers that predict risk of age-related cognitive decline that can be implemented in community or primary health services to empower individuals to map their healthy aging journey. And I think that's what Frini is going to talk to us about today. And Professor Karayanidis is an elected fellow of the Society for Psychological Research and the Association for Psychological Science, and has been awarded a Lifetime Achievement Award by the ACNS. So, Frini, thank you for giving me today.
Thank you for inviting me. It's fantastic to see people. I would also like to start by acknowledging the Wurundjeri people of the Kulin Nation, and as well as the Awabakal and Worimi people, who are the traditional custodians of the land that I come from, and pay my respects to their elders past and present. And I also just want from the start to acknowledge the team that has been behind this work. This is the current version of my team. It's a little bit pared back, but that's it's a strong group, as well as the collaborators at the University of Illinois. Some of the foundational work that has led to the work that we're doing here is still happening at the University of Illinois Urbana-Champaign, as well as Ash Smith from the University of South Australia, with whom we got the initial grant that collected most of the data that I'll be presenting. Now, I've got some introductory slides that I know are probably unnecessary, but I thought I'd leave them in just to put us in the mood for why we do this stuff, as opposed to inform you about the needs for healthy aging and the healthy aging agenda. So, you know, we all flash, colours are a bit funny, but we all flash this snake graph from Livingston at any talk about healthy aging and dementia risk prevention. And every 3 or 4 years, we get increasing number of risk factors for dementia risk, with now up to 45% of factors being potentially of contributors to dementia risk being potentially modifiable. And we also have, you know, again, I'm not going to read through this. We know that we have an issue. I don't want to call it a problem, because I think healthy aging and increase in aging population is good. It's what we do with it that is potentially a problem. So essentially, I think it comes to us to this need for prevention approaches and giving people agency about how to improve their healthy aging journey. That is the challenge, rather than the challenge being that people over 65 will double in the next 40 years. Now, for those younger amongst us in the room, this is you. So people in their 20s, late 20s, people in their 30s and 40s, these are the people that will increase our aging numbers in the population. Now, and again, this is no big revelation, but we need. Do you want me to press 4 out? I always do that and then it comes back again in an hour. Just to annoy us. So we all talk about aging biomarkers. I remember as a PhD student, the P300 of the related potential was going to be the biomarker of dementia, right? And we've looked at genetic and clinical and a whole lot of other things. And I think the problem is that we talk about a biomarker, or even biomarkers, and I think what we need to work towards is composite biomarkers, using machine learning, using all the knowledge we have to develop something that is sensitive enough. But I also think that the problem is that we sort of do it as we want to assess you once and then see what your risk is, and then put you in the you are at risk category. Whereas we know that, aging is not a singular pathway. This represents two pathways, there's more. And within all of these, there's lots of different lines and levels. From my perspective, what I think we need to do is to figure out how we can be assessing people over their life longitudinally in clinical practice, in GP clinics, in such a way that we can then identify a drop, identify something that's concerning enough, just like we do with hypertension. You go a couple of times, hypertension's, your blood pressure's high, they say, come back in 3 months, we'll test it again or monitor it, and then we will put you on medication. Something like that, but with a bit more sensitivity, because by the time you've got hypertension, your vessels have already started having a problem. So our measures are not sensitive enough, and they're not, other than things like blood pressure, are not particularly suited for longitudinal assessment. Not surprisingly, a lot of these risk factors in midlife are associated with cardiovascular, cerebrovascular health, high cholesterol, physical inactivity, diabetes, smoking, hypertension, obesity, alcohol. And they're all modifiable, they're preventable with lifestyle changes and medication, some of them with medication, some of them not, but all of them involve to different degrees cerebrovascular changes that occur at the level of the arterial wall. So whether it's atherosclerosis or arteriosclerosis, or a combination of those, as they often occur together, together with other factors. But here I'm focusing more on arterial health. Coming back to blood pressure measurements and other conventional assessment methods, they generally represent accumulated damage over years. So by the time you're diagnosed with hypertension, your blood vessels have already started changing or have been changing for a while. And so essentially, again, my argument is that prevention and early intervention requires monitoring of some more sensitive measures over time to identify a point of drop. Most of them, oops, that was enough, it seemed. Okay. Most of our measures are peripheral. What is available at the moment, I mean, we've got brain measures, too expensive, giving everyone an MRI scan every couple of years, not feasible, and are based on the arterial pulse wave. As we know, arterial blood is pulsatile, it moves forward, and that pulse is reflected back when it reaches the endpoint, and it produces this pulse wave signal that then is what is used for our systolic and diastolic blood pressure measurement. But that pulse wave changes, these two, the forward and the reflected wave, changes as a function of arteriosclerosis, resulting in a difference in the shape of this pulse as a function of diabetes, hypertension, age, and other factors. But these are measurable components of it. When we look at measures of pulse wave, I guess pulse wave velocity is the gold standard, taken with the carotid femoral measures. Sorry, the slides are not, the projection is not very clear, but this is a peripheral measure of how fast the pulse travels from the femur to the arm, yes, to the carotid artery. There's carotid femurs and zanchial, brachial ankle. But these measures in studies have shown quite consistent drop cross-sectionally in global cognition, in executive functions, in memory, in a range of studies, as well as longitudinally, as you can see here in these measures as well. The American Heart Foundation has recommended that pulse wave velocity be assessed as part of regular clinical practice to identify a high risk of cognitive decline or early stages of decline, but also heart changes, right? Risk of heart disease, risk of stroke, and so forth. But from, I don't know why it likes to do that. It must be doing something funny. But from the point of view of what we're measuring in terms of sensitivity to cognitive decline, this is a peripheral measure. So we're measuring something that is happening peripherally and most possibly becoming evident years after mild changes have happened at the brain level. So this is where the work from, faces, of Monica Fabiani and Gabriella Gratton came in. They are were the early developers of EROS, the event-related optical signal, which is a fast optical signal coming from the near-infrared spectroscopy, but with systems that allow you to have resolution of temporal resolution of 1 millimeter and very good spatial resolution. But what they started doing in the early 2000s, or mid to 2010 and so forth, is take that measure that they have with these helmets. You have sources of light, near infrared light and detectors. So it's the same as the near infrared spectroscopy. And depending on the distance between the source and detector, this banana shape can penetrate into the surface of the brain or not. So essentially, just like with EEG, when we process to get event-related potentials, we get the horizontal and vertical eye movements. We remove those as artifacts and throw them away. Typically, in near-infrared work, this signal here, the cardiac signal or the arterial pulse wave, is removed as artifact. So they actually process that artifact. And so they're looking at, I'll just put it up together, the change that is happening in this pulse wave as a function of age and other conditions, so that this signal differs between younger and older arteries, which are stiffer, because the forward and backward wave come back closer together. And that arterial stiffening is, over time, produces these additional changes, cumulative changes in reflected waves, pulse wave, pulse pressure, and pulse wave velocity. So the type of measures that they developed are very similar to pulse wave velocity measures, but in addition, they looked at the shape of this arterial wave. So you can see that's the timing of the peak systole versus the timing of the peak diastole. You have pulse transit time when it's time locked to the cardiac pulse. Then you have the amplitude, peak amplitude being the amplitude of the systole to baseline. And they patented a measure of what they call pulse relaxation function or prefix, that then looks at the shape of that wave. Now, this is the way that they measure it. Just put up the next slide. So you have, I'll emphasize this because that's what I'll be talking about today. It's the sum of the forward and backward pressure waves, and it depends on the speed of the recoil between the systole and the diastole. And it is normalized to reduce the effects of age that you have on the other two measures. So they've shown, I'll give you some information about a brief review of their work, that it is related to compliance or arterial stiffness at the point of measurement. So what they also did was record from the whole cortical surface or cranial surface, and then map that, co-register it to the structural MRI, and they can get an image that looks like an MRI heat plot, indicating regional variability in, this has gone to sleep. Regional variability in prefix or elasticity across different regions. This is one of the earlier graphs that they had, where you can see people with different ages, so 29-year-old to 70-year-olds, and different levels of cardiorespiratory fitness. The level of I'll define it in more detail later, but essentially a measure of cardiac fitness. And you can see differences between them in overall arterial elasticity or prefix, across people of different ages, but also even within the same age group as a function of how fit you are. So people with less fitness show lower arterial elasticity than people with higher fitness. So they've shown that prefix varies with age, with cardiorespiratory fitness, with lower whole brain or regional grey matter and white matter volume, with lower frontoparietal functional connectivity measures, and poorer performance on executive function tasks primarily. A more recent paper by their group, by Daniel Bowie, always gets me, is they showed that in a cross-sectional sample, that they looked at the break point for prefix versus white matter lesion, and you can see that this break point happening just around this interval here is about 8 years earlier than the break point for white matter lesion load. So it's an interesting finding that shows that it's a bit more sensitive to changes than white matter lesion. So they developed this framework, which we are working on as well, to try and understand these relationships. Hi, Kristen. Across the different levels of the system, and arguing that, oopsie, I do. I don't know I do. I don't think I do. That cerebral arterial elasticity may be an early indicator of changes that are happening further down the track. The work that we're doing together for the last 5 to 6 years is looking further into the effects of cardiovascular health or measures of cardiovascular health on age, which we know exist, or the interactions, how age impacts cardiovascular health, but also looking at the relationship with other levels of this cascade, but also looking at lifestyle factors, how they impact cardiovascular health, how they impact this cascade, but also looking at particular components at each level of this cascade. So I'll present to you a little bit of work from that. The work that we're doing, this is coming out of 2 studies, the Activate study, which is run by, was awarded to Ash Smith in UniSA, and I'm the lead at Newcastle. We also got funding for the ABC study, which is, this is the first one. Activate is a longitudinal observational study. ABC is an intervention study. I'm not going to be presenting data from here today because I presented a bit at ACNS, and it's also now we're doing more rigorous analysis, and there's not enough time. Please keep me informed of the time. I don't have anything. Fantastic, thank you. So this is just to orient you with the Activate cohort generally. So that we are using in the studies that I'm reporting. This is the largest cohort, so I've presented it here, but some of the other work, because we can only measure the optical signal in Newcastle, whenever we're using the optical signal, it goes down to about 200, 180, because that was the size of our sample there. What is important to notice is that these people, even though they were recruited during COVID, are really healthy, and probably because they were recruited during COVID are really healthy, because those were the people who felt comfortable coming out of their homes, and every time the lockdowns stopped, they would come and do testing. They're cognitively very well. All of their markers there, the MVPA is higher, is 90 minutes a day, 90 minutes per day, yes. It is much higher than expected. And all of their markers seem to be in the healthy category, but even so, when you look at their level of whether they do have hypertension or not, or whether they're medicated with hypertension, you can see it's about 50-50, and the same, a third did not have cholesterol, high cholesterol, and 2/3 did, based on blood measures, but also based on their report of whether they have been diagnosed with hypertension or hypercholesterolemia and so forth. So this is the first study that I'll talk about today. This has been, it's currently under review, but it's also on bioarchives, where what we wanted to understand, this isn't using the optical measure, this is trying to understand whether cardiorespiratory fitness versus cardiometabolic health affect the same or distinct cognitive domains. And this is the overview of the model that we used. We used about 350 people to develop a model of our main measures. I'll explain what they were in a second. Then look at the effect of cardiorespiratory fitness on these 4 domains, cardiometabolic health, and then their joint effect when controlling for their common variants. I'll put these both up and it'll be easier. So cardiorespiratory fitness refers more to a functional state, the capacity to perform sustained physical work, involves the integrated function of different health systems, skeletal, muscular, skeletal, respiratory and cardiovascular. And it's VO2 max would be the gold standard, but it can also be assessed using non-exercise algorithms that take into account a number of cardiovascular or lifestyle factors. So typically these non-exercise algorithms include age, sex, BMI, resting state, and self-reported physical activity. In our case, we used BMI, heart rate, and recorded MVPA over average over a week. We didn't put age and sex in there because we're controlling that in the models. And then with cardiometabolic health, it's more of a structural, if you're like, or a vascular and metabolic integrity measure, a more consistent measure, so you can have high levels of poor levels of cardiometabolic health, but still have high level of cardiorespiratory fitness. And here we used blood measures of glucose, HDL, triglycerides, BMI as well, and forewarning, it didn't really matter whether BMI was in there or not, so I can discuss that later, but it was contributing to both factors, as well as systolic and diastolic blood pressure. The cognitive domains were made from a number of different measures, and you can see here both measures of NIH, from the NIH Toolbox on Education and Crystallized Ability, verbal memory, recognition primarily for verbal memory, executive function, the multitasking task, but also acute trials task switching paradigm. We used 2 scores that are equivalent in terms of across the 2 tasks, but refer to interference control and task switching cost. And 2 measures of reaction time, simple and complex, from the reaction time task at the Cantor. This is how they loaded, so it was a pretty well designed and stable model. Now, when we look at cardiometabolic health, interestingly and unexpectedly, It loaded onto crystallized ability and verbal memory loaded onto cardiometabolic health. Whereas for cardiorespiratory fitness, executive function and processing speed loaded onto cardiorespiratory fitness. Interestingly, you can see that the other two pathways are non-significant. Now, these coefficients are in a similar range, but they were not significant. And you will see later when we put them together, the values are lower still. So when we put these together, we found that, yes, indeed, these two do correlate. They have quite a substantial relationship. But the pattern of relationships with the cognitive domains was identical. And in fact, these became much stronger, 0.31, 0.28, and especially the executive function one became much stronger again. So controlling for the association between cardiometabolic health and cardiorespiratory fitness retains this double dissociation between cardiometabolic health and more crystallized, consistent abilities that are less impacted by healthy aging versus cardiorespiratory fitness impacting abilities that are much more sensitive to aging, to healthy aging, not with the dementia risk. In this next study, this is now with prefix, and so it's a smaller sample. It's 163 people from Newcastle. And initially, because all of the work with prefix has been done at Illinois, we just wanted to see, can we replicate the patterns that they find? And then can we also understand the relationship with cardiovascular health, indices, and global cognition? As you can see, our sample, in this case, we just use a dichotomous. It's more of an assessment of the status at the moment. Is there systolic blood pressure in the high range? Is there cholesterol level in the high range, glucose and BMI? And we also looked at their total cognition or global cognition of the Addenbrooke using, which again, you can see they were all very high, with no indication of any cognitive decline. And we measured prefix. We have a much smaller system than the Illinois group. So we measured prefix in 3 consecutive montages over focusing over the left middle, the areas, frontal areas, and a bit of temporal and parietal perfused by left middle cerebral artery, right middle cerebral artery, and then the midline with anterior cerebral artery. What you can see here is the sort of the spread of prefix measures when averaged across all regions, and this is for particular regions, and they were very highly correlated. We also took measures at 2 3-minute intervals, looked at reliability over 95%. It's a very reliable measure that you tend to get because it's a very large measure. It's like the EOG, the vertical eye blink. It's a very large measure. It's pretty reliable. And then we looked initially at relationships with different, these are just simple correlations, with different indices similar to those used by the Illinois group. You can see with age very consistently, and different areas showing different levels of significance, but essentially the scores are in the same range. Not with ACE, which is not surprising given that our participants were very high functioning. Strongest with pulse pressure than with either systolic or diastolic, with heart rate, HDL, cholesterol, glucose, BMI, and that measure of cardiorespiratory fitness, as well as grey matter. So across the board, most of these results are replicating what has been done in Illinois, and this was providing some new evidence of relationship with cardiovascular health measures. when we looked at the number of cardiovascular health measures on which people which were present, regardless here of medication, you can actually see that people with no cardiometabolic health or cardiovascular risk factors have higher prefix and significantly higher than those with 3+, and the same people with 1 are significantly higher than people with 3+. So this is the first time that we've seen this type of relationship. Here all of this is still in preparation, so we haven't actually looked at which ones are producing this, is it level dependent and so forth, but that work is currently underway. When we just visually to show you these 3 montages for people with no cardiometabolic health versus 3+, you can see quite a large difference in arterial elasticity visually, which is producing these group effects there. So in terms of cross-sectional information here, we asked, can we replicate? And yes, we found that prefix is behaving the way it's expected in terms of other demographic variables. It's not predicting total ACE, so total global cognition, and it doesn't predict global cognition even when we control for the number of metabolic health factors. But I guess that's not surprising. These are, as I said, ACE, they're performing very highly, and these are pretty crude way of classifying cardiometabolic health. I won't go into this just to save time, but essentially it's saying what I said before. We then looked for the first time in a smaller sample. So this is at 1.5 years. Activate had baseline 1.5 and 3.5 years. At that point, we still haven't processed the optical data for the 3.5 years. It just finished recently. But what you can see is that there was, interestingly, actually, there was improvements in almost all of the cardiometabolic health measures, except for heart rate. So being in the study made them more aware and made them become more active and take care of their health. We don't know, but at least these people did improve. This was a small subsample that did these measures.
Can I just ask, was the first phase during COVID? Then?
Yes, the first phase was, and could be stress related.
Or moving less.
Or moving less, absolutely, yeah. Even though you sort of MVPA levels were pretty amazing. They would come to us and say, oh, I just went rowing at 4 o'clock in the morning, and then I did a half marathon, and then I'm here. So, but even so, it was interesting to see that there was a decline in prefix over this small interval, a 7.9% decline. If you look at overall statistically significant, even though the interaction, was significant, you can see that the drop was statistically significant for right MCA and ACA. So there was a main effect and a time by phase interaction. So that was interesting to see that decline is happening even though there's not evidence of much happening there. When we looked at, again, these relatively coarse measures of cardiometabolic health, there was no relationship between their presence or absence on the amount of change that happened. So again, that's interestingly, excitingly, the first evidence of a decline, but we need to do more work to understand the factors behind that. And probably we need the first phase of data so we get a larger sample again, because this is fairly small to do this work. The next study that I'm presenting is looking at regional associations between cerebral arterial elasticity prefix and grey matter volume in healthy older adults. That work is following up from Chiarelli's work, which is with the Illinois lab, who found that in a lifespan sample, a relatively small sample, that reduced prefix regionally was associated with lower grey matter volume at the same region. and that the associations were much stronger frontotemporally in the older subset, but this was only 28 participants. So we now have 162. We have, that's why I didn't emphasize, this is a restricted age range of 60 to 70, which we deliberately did to remove these huge effects of age that happen in that age range, but that also still happen in 60 to 70. We didn't manage to eliminate any age effects completely, they just reduced. And we're looking at age prefix and grey matter volume relationships, and the variability, whether variability in prefix is related to cardiorespiratory fitness. So this shows you the figures from prefix across the areas that we can record from, or we recorded from. The left MCA areas, the ACA areas here, and the right MCA areas. And you can see that there's substantial variability between regions. Again, lower values, especially around anterior areas, and some, well, higher values, sorry, lower values here and higher values more posteriorly. Looking at the relationships between age and prefix, even in this narrow age range, they were significant, and age correlated with prefix significantly, primarily across anterior prefrontal areas. And then what we did is take the cardiorespiratory fitness score. This is the estimated cardiorespiratory fitness from the JERCA estimation, and used that to create low fit for age and high fit for age groups, because even in this small age range, we had an age effect. So we just did a median split, got low fit for age, high fit for age, and you can see that across all of the areas, to some extent, people who are high fit for age have higher prefix than low fit for age in both hemispheres in most areas, but the ones with the asterisk were statistically significant. Again, primarily across prefrontal areas, but some temporal areas as well. This is what I showed you before, the age by prefix. We also looked at age by grey matter volume. And again, you get large age effects even in this restricted age range. But if you eyeball the colors, there's some level, they're not the same values, but they represent a similar range. Again, there's some level at which the relationships with age are larger or more consistent, if you like, across frontal areas. And then we looked at the relationship between prefix and grey matter volume, which again have their strongest associations over lateral frontal areas, and especially across this area that merges middle frontal and inferior to the superior two parts of the inferior frontal gyrus. So we wanted to look at whether prefix mediates that relationship between age and global and grey matter volume. And we also wanted to look at whether it's moderated by sex, because every time we would look at sex effects, they're there. Women have, well, first of all, we had 60% women, 35% men, but also women had higher grey matter volumes, tend to have higher prefix, tend to have higher, better cardiovascular health generally. So we used this ROI that showed the strongest relationships between prefix and grey matter volume. And we looked at a mediation model with sex as a moderator. There was evidence of a moderated mediation, and the model was significant, with, as you can see, significant pathways across all three pathways, significant findings of significant beta values across all 3 pathways. And then when we look, sorry, the colours are difficult to see. When you look at those age effects, then there is a significant prefix by sex interaction, which shows quite clearly that the effects are stronger, much more stronger in women. In fact, the beta coefficient, the coefficient is, if anything, a little bit on the negative side in men. But again, 2/3, 1/3 of numbers. So quite a different effect size there. So conclusions here, prefix and grey matter volume are significantly correlated over this mid-inferior frontal region. And the relationship between age and grey matter volume is mediated by prefix and the relationship moderated by sex. As with the other work, this is still very much in development. We are intending to replicate this with our ABC sample, our other sample at baseline, which is 150 people, and then combine those two, if we see the same patterns, to be able to look at the sex effect in a much larger sample and look at the longitudinal difference. And just finally, I wanted to touch on the final study I'll just very briefly mention today, which is starting to look for the first time at relationship between this measure of cerebral arterial elasticity and lifestyle factors. In this case, it's the work of Felicity Simpson's PhD, looking at whether Mediterranean diet can predict or is associated with cerebral arterial elasticity, and whether that relationship is moderated by activity level and energy intake. 159 people in this case, the same. This is now a single frontal cortex ROI, prefrontal cortex rather, ROI, and med diet score was based on the Australian Eating Study. We used 3 regression models, multiple linear regression models. We're very happy to find that med diet predicts prefix. The effect remains when controlling for age, sex, and education, especially education because it can affect dietary patterns, and also when including total level of activity from MVPA and total energy intake. So that was a very pleasant finding, again, just to show you the lowest quartile on med diet adherence versus the highest quartile, showing quite different patterns over this prefrontal cortex mask. So this is the first evidence of a link between dietary patterns and regional cerebral arterial plasticity, and again needs more work to be done, and longitudinal work to see how that changes and whether prefix also mediates the relationships between dietary patterns and functional structural brain changes. So that is all I'm presenting today. I know it was a lot and I know I speak fast, so I hope I haven't bamboozled you. And I want to thank my team again, but also to let you know that there is a PhD scholarship opportunity with the next phase of the study. We just got funded last year for extending our ABC cognitive intervention to look at the joint effects of cognitive and physical intervention. So what we're hoping to do is to see that the peripheral physiological arterial changes that happen with physical activity can enhance the effects of cognitive training that are happening locally. So thank you.
Very much for your talk. Any questions?
Thanks, Dr. Frini, for a very fascinating talk. My question is, did you find any confounders or any other variables that might affect your results in your study, or is
I guess in different studies that are presented or different papers that are presented, there would be different types of potential confounders. Probably the easiest one to look at is in this instance here. these could be potential confounders of that relationship or factors contributing to that relationship. Generally, we do include age and sex in all analyses, but we don't put it in the estimates of cardiorespiratory fitness. We control it at a variable Y level. Are you thinking of anything particular?
Could be backgrounds or anything that, apart from the potential confounders that you.
Look, background, Newcastle is not Melbourne. It's extremely homogeneous. So I would say that 98 to 99% of our participants were white Anglo people.
And do you have any like income, to just follow up on that, do you ever include income type measures? So you've got education?
In the very first, I won't flick through, we only got a socioeconomic level based on postcode in this study, and and that's not a very good measure because it's a population wide, it's the whole, and also again in Newcastle, you can get in the same, in St Kilda, for instance, you can get very high income families and very low income families coexisting because you've got, community housing or social housing. But they were in the above the average for the region.
Thank you for the talk. I'm going to ask a question about one of the results in the first study, which was, you don't need to flick back either.
No.
There was quite a strong association between, I think it was executive functioning and cardiorespiratory fitness.
Yes.
And I was just kind of curious to get your thoughts on the direction of causality, if there is one in that relationship. Do you think it's possible, for instance, that people with higher executive functioning might be more likely to engage in healthy lifestyle behaviours?
Well, we interpret it differently. Sorry, I thought I'd get to my previous slides this way, but I lost the picture there. That's okay. In that slide, because what we're finding is cardiometabolic health is associated with crystallized ability and verbal memory. Now, these are much more stable abilities and affected by education level, by ability level, this foundational ability. Thank you. So we're arguing that that relationship may very well be level of health information, health literacy, looking after yourself and your cardiac health. The other one is just a little bit more difficult to explain that way, especially because cardiorespiratory fitness didn't also relate to crystallized ability. So it seems that the interpretation we're putting in that paper is that, in fact, that's likely to be the lower your arterial health or your cardiorespiratory fitness, the lower that is starting to affect your executive function, whereas the other one might be reverse causality, that the crystallized ability, your level of functioning, global level of functioning is affecting how well you're looking after yourself. But yeah, these are cross-sectional correlations, essentially.
Can I follow up on that? Were there strong correlations in performance between the crystallized ability, verbal memory, and executive functioning and processing speed? Because you could be...
No. How can we go one slide up? 2 slides up. Sorry. If you can put it into presentation mode. Sorry. So you can see, executive function was associated. I'm checking that it's all positive associations because here we've got all the variables positive is good.
Yes.
And with verbal memory, not processing speed isn't. So yes, there was associations at that part of the model. Look, I really think that this, as soon as we can, we need to look at it in the longitudinal data set. That will give us a much better ability to make some reasonable predictions.
I've got two questions, but one is, depending on the results, some of them were specific to certain brain regions and sometimes it wasn't specific. And I just wonder, why would you have specificity in terms of if it's, why would vasculature in certain parts of the brain be more affected than in other parts of the brain?
Well, what I didn't emphasize is that across the papers, we use sort of larger masks or whole frontal cortex or whole prefrontal cortex versus more regional. And the nutrition one, for example, was total prefrontal because we wouldn't expect any regional. Why would nutrition affect your inferior frontal gyrus more than your superior frontal gyrus? So in those cases, we're not using regional, we're not examining regional effects. In the third paper that I presented with the
even the prefix versus age, it seems that in different parts of the brain, it was a different frontal cortex region, it was slightly
Yes.
different. And
Yeah.
I'm just wondering why would that be?
Well, we do know that, yeah, that's a good slide to put up. We do know that age effects vary across in volume, in cortical thickness, in cortical atrophy. They vary across regions of the brain.
But for your prefix measure,
Well, if we think of that cascade of what is happening at the end, if you think of grey matter volume or white matter lesions as sort of a further down pathway in that cascade, it may well be that the reason you're getting that damage in frontal areas is because frontal areas are starting to be less efficiently perfused.
yeah, but it's
Now,
just so
why that would
yeah,
be,
why that would be.
I don't know.
And I've got another question about and I have the same problem in development or participant sampling. So how do you account for the fact that you've got this biased sort of quite healthy aging group with more female than male? I mean, how, when you try to extrapolate your results, how do you?
Absolutely, Roz. It's my bugbear. And I think the way we do it is to take this stuff into community. I mean, who comes to universities to be tested? Even in a multicultural community like Melbourne, what's the proportion of your white mainstream participants, except for students, right? Not student studies, because that would be much wider, but then you've got the highly educated versus the more mainstream. I think we're doing the community a disservice. In my other life, I'm working on a different project, which is it's called Community Healthy Aging Initiative, bringing information about healthy aging into the community, but also eventually bringing measurement of these things in the community.
Because your setup is portable, I guess.
With this can be done with a small near systems. You won't get the regional specificity, but this can be done with small near systems, 10 minutes in a shopping centre, in a health clinic. That's what if I live long enough and am employed long enough, who knows? My aim is to be able to develop this measure with enough confidence behind it to say, pulse wave velocity takes a good half hour to measure and it's only peripheral and you need specialised people. This, with the portable systems, you throw some diodes on, you record it in 10 minutes and you can have every couple of years, just like our blood pressure is taken and we do our health checks.
A question just on that translational note about the implications of your findings for the community. With the double dissociation with cardiorespiratory versus cardiometabolic health, what do you make of what we should do with, or what people should do with that information in terms of their lifestyle? Because often they're sort of lumped together, and you talk about, improve exercise, diet, lower your blood pressure, all of that lumped as one thing. Do you think, given your findings, that exercise is disproportionately important to the things that will change or more likely to change over time cognitively?
This is an extremely speculative answer. But if what we saw between the more crystallized abilities and cardiometabolic health and cardiorespiratory fitness and executive function, if this is true and has to be, we actually have an honours student replicating that in the ABC data currently. I don't know what the outcomes are. Then I would argue that for the cardiometabolic health crystallized ability relationship, that reflects health literacy, could reflect, right? Speculative completely. So in that instance, you need to inform people and have a proper system where people are aware of the importance of whatever it is that makes us our cardiometabolic health, our risk factors. So that can be done at a clinical practice level. Because a lot of people don't know. The other thing that frustrates me, when I go into community to talk about, you know, healthy aging events or seniors weeks or whatever, half the people in the audience are my participants. So preaching to the converted.
It would also reflect just socioeconomic status and
Absolutely.
ability to not just to know that information, but to implement it.
Yes, absolutely. But, you know, we've got a pretty good healthcare system with Medicare. We could do more.
It's on the preventative side, I suppose.
Yes, exactly. But the other relationship is more, seems to be more related to the activity, lifestyle, you know, which would still impact your cardiometabolic health. I mean, they're related, right? The coefficient was 0.55. They're not independent by any means. But I think the message is positive insofar as saying you can approach it, if the model is correct, you can approach it in different ways. You can improve your cardiorespiratory fitness, but you can also improve health literacy, which we know Australian health literacy is low. It's very low. I can't remember the numbers, but.
Thanks, Frini. I really enjoyed this. Biomarkers have been a bit of a holy grail in neurology and psychiatry for a long time. And it hasn't been a lot of success because largely you're trying to identify, especially with cognitive and neuroimaging measures, something that has a combination of high construct validity, high test-retest reliability, and discriminability in terms of identifying people who are at risk. And often that combination is hard in these measures because they're so multifactorial. So I'm just interested in how your approach might differ from some of those in the past.
Well, to be perfectly honest, it may or may not, right? I mean, but I think one of the things that's missing from most biomarker research is repeatability, is multiple testing. And so we're trying to look at people cross-sectionally, and we know that variance is all of these other potential confounding factors, including the fact that we don't get the work. I think what we're studying across the board, not for us, but in different places, different amounts, is the cream of the crop. So we don't really know that variability. And so.
But do you think that this is the wrong word, but the simplicity of the cardiovascular measures are in that they might be less multifactorial in what's driving change?
I think so. It's like, again, I'll put the analogy with the vertical eye movements. It's such a big signal. It's, you know, it is a quite consistent signal. We saw a change, but it wasn't a huge change, and there wasn't much in terms of we need more work. That was only 64 people over 1.5 years, but there's quite there's consistency and variability in that measure. It has very high internal reliability. So if you test it, as I said, over those periods, but I also don't think this is going to be the measure alone. And I think because something like this could be measured in clinical practice relatively regularly. I'm even thinking you put up a stall in a shopping centre. Everybody who goes by, you get basic information, systolic blood pressure. You come, and we've been doing that, not with this measure, but with, you know, just like come and do a health check. People love it. And that's where you get also the people that are not those very high functioning coming to do it. And then if you combine it with things like pulse pressure or heart, cholesterol levels, and all of these other biomarkers or markers that we have of health, and you follow it through over time, perhaps with genetic information as well. I guess I don't want to, I cannot promise, you know, we're speculating here, but it's a good measure that could be used.
Okay, great. I suggest we continue discussion over food. Thank you again, Frini. Very coherent set of things. It flows quite well.
It's difficult with this structure.
I have until like.
It's hard to do this work.
Yeah.
There's not much other research out there with these measures.
Yeah, I was pretty sure that was.
Yeah,
okay.
yeah. Well, there's only a few groups that are using this.
I walked in and didn't know she was sitting there.
One in Montreal or somewhere in Quebec. These guys have a couple more labs, but with this measure, these are the only treatments. And so even it was important for them to be able to say, similar results. Hey, Kristen, thanks for coming.
Yeah, I guess how do you?
So, Daniel's lab.
Is the Shane Warne testing of people going to